From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on October 17th, 2025. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 113th episode of Matters Microbial. I am so very grateful for every listener, viewer, guest, and supporter. Truly. The first thing I want to do is mention a wonderful microbiologist about whom I insist my students learn. The late, great Dr. Esther Letterberg. She was instrumental to the early days of microbial genetics and was heavily involved with the research of her then husband, Dr. Joshua Letterberg, a Nobel Prize winner. Sadly, Esther did not get the credit she deserved in those early days of microbial genetics. I'll put a link to her many accomplishments and fine perspectives in the show notes. As I insist to my students, if you do the work, you should receive the credit. I was lucky enough to get to know her while I was in graduate school at Stanford in the 1980s, and this photograph is how I remember Esther. My wife, Dr. Jennifer Quinn, created this wonderful Lux portrait of Esther in her younger days, painted with luminous bacteria. I love that smile. Jenny went on to 3D print this glow-in-the-dark memento of Esther that I believe would have made her happy. I know it makes me happy. Please read about Esther Letterberg and her many central accomplishments, including things like phage lambda. She was a friend of mine long ago, and I miss her. My Bio 350 Micronauts had quite a week, to be honest with you. We're looking for microbes that produce antibiotics from moss and lichen samples. As you can see here, moss samples produce a wide variety of microbial colonies worth considering. In this plate, I have pointed out areas on a crowded dilution plate that appear to have some antibiotic activity. We will certainly test the associated colonies in a couple of weeks for their activity. Again, these are student isolates from samples they collected, and that makes a difference to them and to me. I also showed my Micronauts the role that environment can play in gene expression. Consider this plate of Serratia marcescens grown at room temperature. The colonies are brick red with the fabulous pigment prodigiosin. I just love that name. However, this same organism, when grown at 37 degrees C, prodigiosin is not made. One of the enzymes in the pathway creating that pigment is sensitive to that higher temperature. Folding issues, I would imagine. After my students photographed those plates at 37 degrees showing the white colonies, I had them transfer the plates to room temperature and incubate for several days. And this is what they see. I asked the students to think about what this could mean, where there's red around the border of every colony, and we came up with a thought, a hypothesis, that prodigiosin synthesis only occurs in growing cells at room temperature, and the borders of colonies contain those growing cells. This kind of student learning is a valuable skill and very important to me. We then ran some of our colony PCR samples from our marine bacteria on an agarose gel. As you can see here, the proper amplicon appears in almost every lane, and I was so pleased to see it. So were they. However, this particular professor then dropped the second gel on the floor, which, as you can see here, made it difficult to observe which lanes had amplicons, though many did. Think of it as a jigsaw puzzle with Jell-O. It's been a lot of years since I've done that, and I'm kind of sad about it, but that's how it goes in science. It's the in research. In any event, off for DNA sequencing. Finally, my Micronauts had an exam today. Part of it was take home, and I asked them to create a thoughtful and humorous meme of some concept in class that really interested them. Here are 3 examples. As you can see, one micronaut thought of Keanu Reeves in The Matrix as a metaphor for how a biofilm can protect against antibiotics. Another student decided they were very, very interested in microbes that preferred very low nutrient concentrations, oligotrophs. And finally, you can see a third student was amazed that bacteria and archaea have something very like organelles, where specialized functions occur, just like our own eukaryotic cells. These personal creative approaches, I find, really help with concept retention and student enthusiasm. Also ownership. Creativity helps and does not hinder learning. One of the central concepts I repeatedly emphasize to my micronauts is sociomicrobiology. This refers to how microbes communicate with other microbes in various ways. Now, this idea leads us to cooperative movement, biofilms, and even quorum sensing. So I am extremely happy to introduce Dr. Joshua Shrout from the University of Notre Dame to the podcast to discuss aspects of sociomicrobiology that his research group investigates. After all, as his website openly states, we research bacterial behavior. Welcome to the Quality Quorum today, Joshua. It's a pleasure.
Thank you, Mark. Great to be here.
Oh, I am so excited by this. By the way, listeners and viewers, I have made a link to, of course, the website that Dr. Shrout has, but also to a particular YouTube video that's part of that site that explains what his lab is about. And even if you're a novice micronaut, I think you'll enjoy it. I know that my students did, and I do. So I think a good way to start, if you don't mind, Joshua, is telling the listeners and viewers your definition of sociomicrobiology.
Yeah, I have a pretty loose definition of what qualifies as sociomicrobiology. I'm willing to accept a version of that definition of anytime microbes are interacting with each other. So certainly this quorum sensing absolutely applies, and maybe that's some of the original basis for coming up with a working definition like sociomicrobiology. But, as one considers discoveries both old and new, we realize that microbes are interacting with each other, and sometimes, of course, they're fighting and not getting along. But there are so many ways in which they're interacting and synergizing and accomplishing things as a collective. Several decades ago, we didn't consider bacteria that are such simple organisms as having any kind of coordinated behavior at all. So I'm thinking of any of those coordinated or synergistic behaviors as qualifying as sociomicrobiology.
I may be misremembering this, and I'd like you to correct me if that's the case. The first time I saw the term sociomicrobiology was an article by Pete Greenberg. And I don't know if that's the first time it was used.
Yeah, I think that's the first use as well. Right. So that's the early 2000s.
And so the interesting thing is I have a lecture that I give my students, and I've given publicly, I call the heresies of microbiology. And that's because a lot of people think they know what microbiology is. And one of the things that I talk about, from the John Donne quote, no man is an island. Well, we tend to be so focused on the individual microbe when in fact, just like the rest of us, they're interactive. And it's very different because we're used to being very, very granular about things. And now we have the tools to start looking at these interactions.
Yeah, I think that's correct. You combine classic approaches to microbiology of, bacterial genetics is traditionally all about exactly granular behavior, probably of one single species. And then the ecology side is, everything thrown together, but you don't understand any of the individual parts. And indeed, now we have the chance to mesh these two in practical and real and rigorous scientific ways. And it's leading to new avenues of discovery.
I am so angry at the universe that I was not born later. Because there are things that we can do now that I never have dreamed of in graduate school. Just to give you an example, if I take a bacterium and let's just say that I'm selecting it to swim faster in motility plates, and I've done this experiment, if you have a reference genome for the original organism and now you have the one, quote, evolved, and that's what laboratory work is evolving the organisms. I can actually, for about $80 a throw, determine specifically what genes have changes. And that's something I never dreamed of because I was so focused on one gene, one phenotype.
Right. Yeah, the advances are quite remarkable. You think about just the onset to be able to use PCR and design primers and go after any one gene you're interested in. And indeed, now we can do whole genomes for only a little bit more in cost. Yeah, it enables all kinds of things. Yeah, it's very exciting.
And it's really wonderful, but it does, and I'm not saying that I'm worried about it, but I want people to be able to kind of have a microbial perspective. And when I say this, people say that, oh, are you inferring that microbes have intentionality? And yeah, I do, but not in the way that most people mean it. And I think a really good example, and this leads into some of your work, is when we grow bacteria on Petri dishes, we have a really strong idea of how they interact with surfaces.
Right.
But we actually can't get our minds down. I mean, it came as a revelation to me, for example, that there's a very thin layer of liquid on the surface of any agar plate. And it never occurred to me. So when I see gliders or when I see swarming and things that you demonstrate, and folks, again, have a look at just some of the embedded videos on Dr. Shrout's wonderful website. It really was transformational for me.
Well, thank you. Yeah, I still put it often in the introduction section of any paper, certainly any review. Some of my colleagues and I that make sure we're exacting measures of practical physics when you talk about bacteria. I mean, we all know if you grow microbes and you leave that Petri plate or a test tube out on your bench for not just days, but maybe weeks or longer, right? They dry out and there's definitely not anything viable there anymore. But somewhere in between when it was fresh and too old, maybe you could go back and restreak and get something growing. And we know that all these microbes require some level of hydration. So then you go back to lab, I'm going to do a specific assay. I'm going to look for swimming or swarming or anything else, right? There's still that thin, at least a thin liquid layer there. Maybe it's completely confluent with liquid and you're not really even thinking about it. But yes, these microbes all need some liquid around, and usually that's water plus other stuff. But that amount of hydration can affect then the behavior that we get, for sure.
I'm a little lucky in that I've always been interested in what I call undomesticated microbes.
Mm-hmm.
And what that means is that if we continually select, and there's such a wealth of evidence about this, when you cultivate bacteria, I mean, in the early days, if there was a variable phenotype in something, bioluminescence or pigment, people weren't interested in the variable ones. They wanted something specific to study. And I'm not upset about that. It makes sense to me. But that's not how the world is. That's how the lab is. And so when I look at things outside of nature, for example, on the moss plate that I showed a few moments ago, there are gliders on there, and because I've seen a ton of gliders in my career, and I'll tell them that some gliders will secrete a surfactant in front of them, so they make their own slip and slide, and then I'm worried I'm too flippant about it, right? So I think something that would be interesting to talk about is I think most folks listening or watching are familiar with the idea of a bacterium swimming through liquid using a flagellar motor, and that's where you get the tumbles and runs, that kind of thing. What happens on a surface? And that thing leads us to swarming, perhaps.
Sure. So as you go from a completely, undefined liquid layer of top to bottom and you compress it, right? And then you think of if you had the agar plate and then not adding any extra liquid, but there just is liquid there, then classic swarming assays are the motility of bacteria on top of the agar, not within a thin amount of agar as classic chemotaxis or swim assays are done. Or, of course, you could recapitulate those same swim assays, in incomplete liquid conditions, but they're essentially the same kind of motility. So swarming is this motility that again is indeed flagellar driven, but it's on top of some surface in a thin liquid film. And this requires those flagellar genes to be active. And in some cases, different kinds of flagellar genes might be turned on and off. So this was first shown for Vibrio parahemolyticus, right? But other bacterial species don't have this differentiation in swarmer states. So that can be very species specific. So this might be the kind of thing that some of us will discuss. I'll use that in air quotes, discuss, or have heated discussions about at conferences and things like that. But it's on top of the agar as we do it in the laboratory. So I have a video of Pseudomonas aeruginosa swarming on agar that is around 0.5%. And so this takes, just under 24 hours for them to, under optimal conditions, from the point of inoculation, they'll spread out. And under these kind of classic conditions, they're going to make these tendril patterns, right? So they're absolutely fascinating to watch. But they're covering ground, and realize that if you inoculate a plate with a prick of cells or maybe 1 or 2 microliters of growing culture, then they're spreading out to be trillions of cells by the end of the assay. So there's a lot of cells, and it indeed requires a large cell population. This is requiring all of quorum sensing and other cell behaviors that we actually do not understand very well, because we know that how the cells are moving next to each other affects the patterns that we get. And this is happening on time scales of a second or less. So it's not some kind of chemical signal. The chemical signaling part is there, but that component, so it gets pretty complex in a hurry, and we know some of what is absolutely required, but we really don't understand all of what's required to get these kinds of patterns in the lab.
And it's so interesting to me because over and over again, I will hear from, I'm going to say microbially adjacent people, which is kind of a misnomer because we're all intimately associated with bacteria, even if we don't like to hear it. But this business of bacteria being simple, they're not. And then when you add to it what I'm going to call emergent properties of groups of microbes together that are very hard to reproduce, and it makes sense to me. It's the way that we look. It's like the way that we look about ecosystems, where you can't predict necessarily what's going to happen. You can see patterns evolving, but you don't know that you're going to get the same thing the second time. So these growing tendrils, which I've seen in tons of isolates from nature, are just fascinating.
Yeah. There's actually some, if you get a physicist or chemical engineer talking, they'll love to talk to you about how fractals form. First time I showed some of my work to a chemical engineer, like, oh, this is just a 2-phase liquid problem, right? And so, well, I guess that's true. So what's a 2-phase liquid? Well, you think of oil and water as the classic 2-phase liquid problem that any of us who have spent time in the kitchen understand. But then as you get to 2 kinds of fluids that are not so distinct as oil and water, but it's like kind of water and then water with some stuff in it that's this surfactant that Pseudomonas aeruginosa makes, or you talked about other species that they make their own surfactant. That's what's happening. That helps. They're literally making their own surfactant to help aid their movement. Without the surfactant, they won't move the same way. But without their flagella, they won't move the same way. Without their neighbors, they won't move the same way. So it's all those things combined, and that's what adds to it. So the degrees of variation, I guess in a controlled lab experiment way, we should see roughly the same behavior from run to run if we're being consistent about it. But I wouldn't expect to ever see the exact same pattern over and over again.
Exactly right. Incidentally, for listeners and viewers, I've gone over your publication record, And I'd like to know what you don't work on. That would be an interesting question. And the reason that I bring this up is that I have long believed, and you've heard me say this, that we advance by bringing different ideas into our field. And if you look at the history of molecular biology, what transformed the field were a bunch of quantum physicists, starting with Erwin Schrödinger, who got involved with that, Max Delbrück, similar people who were able to bring that mentality into the study of, for example, microbiology and molecular biology. And in a similar way, this ability to talk with physicists about what we're actually seeing is really important. A million years ago, I did some work with atomic force microscopy and how you could measure the consistency of a surface by tapping at it. And it was revelatory to me to think that way, because I was so used to thinking them as these little units that just are not flexible.
Right.
Or as our producer often says, squishy.
Mm-hmm. Yeah. Well, I'm an engineer turned microbiologist. So indeed, I have worked on a variety of things, and even more broadly, have come to microbiology having not done it as the first thing that I got serious about. So in some ways I do it wrong, but in other ways I just do it quite differently and bring a history of perspectives that is, informed how we do science today, both me personally and in my lab. So yeah, a little bit different approach than other of my colleagues who are kind of straight up microbiologists.
I believe I sent you a video of what luminous bacteria do with a time lapse.
Yeah.
And I in fact showed that to a biophysicist I know, and she told me, well, that's just the availability of oxygen and how that turns over in the surface. And there's quite a literature about it, in fact, because, I look at it and I think, I'm looking at all these strange, what look like things moving and they're not. That's where the light happens to be. And this is a great example, again, of bringing different disciplines in.
Yeah. And I bet you're both right? I mean, I bet they are moving, and it's differences in the oxygen, right?
Yeah.
So now you've got two or more variables. It's not just one thing. Yeah. And that gets back to, the kind of the ecology perspective we were talking about it, right?
Right.
I mean, nature and other places where microbes live, right? I mean, that it's complicated. So we're trying to distill it down in the lab to make it reproducible, but the real thing is always more complicated.
Well, I mean, it's like the joke about your Facebook status. It's complicated. Well, that's life.
Yeah.
By definition, right?
By definition.
Right.
Yeah.
And it's funny, we try to impose simplicity onto it, and I would say honestly that life is resistant to that. And it makes sense because of and that's why I talk about a microbial perspective. The last podcast, I was reminded of a friend of mine, Bill Metcalf at University of Illinois, Urbana-Champaign, who told me that phenotypic assays are for our benefit, not the microbes. And we forget that.
Yeah, we forget that. Right.
Yeah. So now we have this information about things moving and making interesting patterns on a plate. And that's wonderful. Now you have interactions between organisms?
Yeah, we can have interactions between organisms. So I think I also included another video we can show that is organisms on a plate, but at the scale where we can see some single cells. And we're seeing how they just, so the video is a time lapse, right? It's fast. It's just a few seconds. But it's from data that was recorded over a few minutes. And we see how these microbes are not going in a straight linear way, and they're not all going in the exact same direction as they expand, but clearly they're influenced by their neighbor or their neighbor's neighbor to some degree. I continue to show this short time-lapse video because it captures the fascination of things I would still like to understand, but we actually understand very little about what leads to the behavior that we see here, right? So I use it to show the basis of why we should do the work, and what we remain yet to understand and discover. But again, the time scale is just, they're doing this. Clearly they're on autopilot to much of what happens. And they're all, probably making the same factors at about the same time. So why are they not all going the exact same direction? I can't actually tell you that.
It reminds me when I would teach genetics, because I'm trained as a geneticist, and you have to kind of not entirely tell the truth to students when you start out, because they believe a genotype confers a phenotype.
Right.
But actually, there are environmental impacts that can lead to changes in what's expressed, and and I love this term, stochastic things that occur. So what you learn, and it's a horrible epiphany for students to understand that the rules that we talk about are more guidelines.
Yeah. If you want really, really tight error bars, you need to pursue work in chemistry or physics, because
Yes.
in biology, often they're going to be a little bigger. I think it is possible to have things reproducible in the lab, and we absolutely strive for that. But, using that word stochastic or other things that we just still don't understand, that is very real, right? There's going to be things for us to figure out. And again, it is complicated. So to chip away at what factors address it, any one behavior that you see can be very difficult.
Well, I mean, if you look at the wonderful work by Jim Hoke all these years ago about the way a flagellar motor is constructed by individual mutants coupled with electron microscopy to find out exactly what's put together over time, is like decades of work, but it shows the delicacy of this. And that's a relatively simple system compared to things like behaviors.
Right.
If you look, for example, and I know you've done some work with myxobacteria, how they do group motility, clearly influencing one another, is just fascinating.
Yeah. Those longer cells compared to other bacteria we know, they don't remain rigid and they get all wiggly like worms as they move. I mean, yeah, I agree with you. It's absolutely fascinating. Again, social organisms, right? I mean, the
Sure.
S motility of myxobacteria, literally named as social motility because it was found that you needed multiple cells in order to show that particular phenotype.
I think it's beyond. I mean, I almost did a postdoc with Dale Kaiser on this topic, and in some ways, I'm sorry that I didn't. But looking at the complexity of the system at that time, I'm glad I didn't.
Yeah, it can be a bit maddening.
Yeah.
But also fascinating for sure.
So I know that several times when I show things that take place on plates, interactions between microbes, and we see them with things we isolate from nature. I have seen antagonistic associations between organisms from reusable water bottles pretty commonly. And I don't mean to imply they're antibiotic, but I have seen that. I think interactions are the rule in nature. Mm-hmm. And we have such a fixation on single colonies, pure cultures, and all the rest. And when I say that, I'm not critical of that. I think it's really important to have a place to start. It's like, you remember the early days of biofilm studies, and people would study one bacterium forming a biofilm.
Yeah.
And then when I was reviewing these papers, I would talk to some of the authors and I would say, well, that's just one organism, and I don't know how often that occurs in nature. And they'll say, well, we've tried it with 3 different types. And I go, okay, does that reflect what takes place in nature? And no, it doesn't. But you can't go all the way over here without starting there.
Yeah.
So if someone were to say to you, are the behaviors that you're seeing only relevant to Petri dishes? You would have a response to that?
I would say that they're at least, they are relevant to Petri dishes, but even what we see in Petri dishes has complexity that we hadn't looked for before. Mm-hmm. And I have an image from a recent paper that we can look at, and it's literally one bacterial colony. And you see the green superimposed on the gray image, and the green are all the live Pseudomonas cells, and then the gray is the whole entire colony. So here is one single colony on a Petri plate, but when we impose some, more rigorous imaging to look at some of what's in this one colony, there's actually a lot of heterogeneity there. So I would use that as part of my argument. Well, even what we've been studying as assuming that all those bacteria within the single colony are completely in lockstep with each other, right? We assume that their genetic lineage is the same, but that doesn't mean that they're all exhibiting the same phenotype. And indeed, now we have evidence that they're not. So some of the areas within this colony where there are no cells are actually dead cells. So some are voids where cells have spread away, but some are actually dead cells. So we'll see these pockets of dead cells within just one Pseudomonas colony. So then we start to scale that up and talk about scenarios, which is what you're getting at. Like, well, how does just one species have to do with what's out there in the soil? And we have some work, limited work, but I still think it's pretty relevant, that talks about how, even if it's multiple microbes that are in a system, if we are considering that system on a centimeters or meters scale, it's just a mixture of microbes. But then if you dig into where are they all, actually they can be quite separated from each other. So sometimes they're actually mixed and touching, but a lot of times they're not touching at all. So they might even be behaving kind of as two separate single culture systems. And this is an area that there's not a lot of work on to understand how close do you need to be to your neighbors to start to feel the effect. And
I think that,
yeah. So I'm very interested in that. We've only done a limited amount of work on it ourselves, but I find it fascinating, and I think it's hugely relevant. It allows for a way to bridge what we've been doing in the lab for decades with other things that we need to do to figure out things we're very interested in, whether it's natural systems like soil or lakes or bottoms of lakes, or infections in various parts of the human body.
we had on a recent episode of This Week in Microbiology some work coming out of Israel that shows that during infections you get pockets of different I'm kind of a splitter rather than a lumper, as the saying goes. And so when you look at a population during, say, disease formation, you can get differences in gene expression. In other words, you start to get different mutant versions of the same pathogenic organism.
Mm-hmm.
And what they're doing is they're adapting to local micro conditions. And that's true in a colony as well as in disease. And this is what I tell students over and over again. We've seen things that we think are easy to study in the lab that you might not think are relevant to a health condition, but in fact they can be. Because it's all about the environment. It's all ecosystems. It can be a petri dish or it could be your lungs.
All about the environment, for sure.
Yeah.
Yeah, we have done a lot of work on that. Going back to my postdoc days, we were just trying to grow biofilms to understand when they would make some of these more classic biofilm structures and when they wouldn't.
Mm-hmm.
And that was then how I got interested in surface motility, because it seemed that they were being cued to be more motile or not under certain conditions. So these conditions related back to basically the carbon source that we used. So if we grew them on more amino acid rich media, they would move around more. Whereas if we just grew them on, glucose or simple sugar, they were more likely to stay put. And then that affects their subsequent behavior as you look at them over hours or days. And we know that amino acids are important for a number of infections, in particular lung infections.
Yeah.
So, this is yet another way in which this one single microbe, in my case, often Pseudomonas aeruginosa, that it can grow in a lake, it can grow in the soil, it could grow on your skin, but it could grow in the lung. So it's not always a terrible pathogen, but when it is a terrible pathogen. But it's a versatile organism in with respect to it'll grow on all kinds of different things, and that affects its behavior.
Oh, absolutely. I don't know if I did have one question. I hope I'm not doing things out of order for you, but you have a wonderful video of what happens over time when two different species are interacting. And it's Pseudomonas, and I'm sorry, I don't remember the other organism.
Oh, it's salmonella, salmonella
Thank you.
enterica. Yeah.
Could you explain what's taking place in this, since it's near and dear to my heart?
Yeah, sure. So we organized the assay. It was a multi-purpose experiment of an experiment, to be perfectly honest. I was making new friends at the university, and one of them was in charge of a new imaging instrument. And this particular instrument is designed to do imaging of both bioluminescence and fluorescence, and potentially like deep, like they could use it for mouse and rat models. And I said, oh, you don't need to talk to me. I'm just interested in bacteria on surfaces. I'm not trying to infect mice. And he's like, no, tell me what you do. So we set up a version of a swarm assay that was not ideal conditions, meaning not the traditional recipe and the methods to study either Salmonella or Pseudomonas aeruginosa, but we inoculated them at different ends of the plate. And, anybody who's done work on motility or particular swarming of either species might kind of know how long it would take their species to spread across the plate. Yeah. And it's no surprise that the Salmonella spreads fastest. So if we'd stop the assay there, you'd say, oh, Salmonella wins. But then Pseudomonas in green in this particular assay starts to take off, and it covers its little area, and then it probably ramps up numerous of its defense mechanisms, and it kills off most of the Salmonella and takes over the plate. But it took 3 times as long as it did the Salmonella to initially spread out. So indeed, this gets to exactly what could happen in a variety of systems, that if you look at any one time point, you're going to think, oh, it's dominated by, in this case, the Salmonella behavior. But it's, the temporal aspect of our experiments, it's a good, food for thought kind of thing, because if we ran our experiments shorter or longer, and then, of course, in nature, they're always being run for, infinite time, right? What's going to happen? It will change. And I guess our catchphrase of the day is, it's complicated, right? So it's always going to get back to that. So as we run that assay out, Pseudomonas aeruginosa takes over and kills off Salmonella on the plate.
Well, it's interesting, and for listeners and viewers, I think that the power of what we can do today just bowls me over. And so the idea of, and I know this is trivial to do for many people, but I really want to emphasize the power of using your brain. And so here we have a situation where one organism is labeled with a particular, I'm going to just go ahead and say in chromophore. I'm sure there are versions of GFP, but the idea is that they fluoresce in different colors, and so you can tell them apart.
Right.
And exactly what you said, people would see Salmonella win, that is to say, cover the plate, and you think story's done, but it isn't. And again, the assumptions you make can actually, and I can think of tons of examples in science where that's happened, where people stopped an experiment too soon. If they would have gone farther, they've seen it. And as you say, it's complicated. They have different agendas, as it were. Now, I don't know for sure what's killing Salmonella. I know that Pseudomonas has type 6 secretory systems. I call them bacterial switchblade knives, but you get the point.
Yeah.
And it's fascinating to see. And this brings me back to the whole idea of heterogeneity in general. Okay? Because if you were to repeat that experiment, you'll get the same result, but the details will differ slightly. Just like if you were recreating an ecology. And that's just 2 members.
Right. That's just 2.
Yeah.
And for me, that's actually pretty complex. We rarely get up to 3 in my lab.
Yeah. Yeah.
Yeah.
Well, I mean, you can start to ask questions of how things interact. It takes us back in a way to myxobacters. But the idea is you knock out a gene and see what the effect on the complex system is. What else are you going to do?
Mm-hmm.
Now, the system biology people tell me they'll get it all figured out, and I hope they do. But as there are so many definitions of that term systems biology, I question that. But life is complicated, and I don't know if I've told you that there's a problem that I tell my microbiology student that the way that we think about biology is eukaryocentric because we're narcissists. And it's also oxycentric because that's what we think about NADH to oxygen.
Mm-hmm.
But even among microbiologists, so many of them are still coli-centric, and they think that organisms, are going to act like E. coli. Not true. Also, the profoundly misguided idea that microbial behaviors are simple.
Mm-hmm. Yeah, I agree with each of those statements.
Yeah.
Some of those we need to try to directly counteract when we speak with other people, and other people we just need to point out there are other versions than the one you learned first. So, yeah.
Yes. Yeah. And I understand why those things happen. And this is how I got called a microbial centrist. Okay. So there you go.
Yeah. I think there's far worse things.
Oh, if I could redo curricula, let me tell you, but they'll never give me that power. It's probably a good thing. I have an unusual view of the universe. I actually have had people say that I like microbes more than anything else, and I'll say no. There are 3 eukaryotes of which I'm very fond: my wife and 2 sons. So there you go.
There you go.
So we've talked about interactions with surfaces.
Yeah.
And we got these beautiful patterns. We've talked about interactions between organisms, often acting in ways that we hadn't expected. I know you have an interesting slide and some thoughts on the idea of quorum sensing, and this is one of the critical concepts that needs to actually be in the introductory curriculum in general, because it's not just about bioluminescence, much as I love bioluminescence.
Yeah. So I have one slide that shows the blue-green of Pseudomonas aeruginosa. So this is Pseudomonas aeruginosa only. And you see in the middle test tube how when you grow it under certain media conditions, most media that we would use in the laboratory, when you grow the cultures up for a sufficient amount of time, roughly 1 day, they're going to make this characteristic bluish green color for Pseudomonas aeruginosa. It's one of the things that is a telltale indicator of Pseudomonas aeruginosa. In fact, so from the clinical point of view, when medical professionals see this on burn wounds and this is never good. But on either side are the test tubes that are missing a couple of genes in different pathways, and those strains are unable to make either the blue or the green pigment that in combination lead to the classic blue-green pigment. And so then so we use that particular slide just to illustrate what we're talking about when we talk about this classic blue and green. And the blue pigment is well known, described since the 1860s, piocyanin. So by this French pharmacist, Fordos, who was describing battle wounds of French soldiers, and he would talk about the blue pus. So that's pio, and then cyan being the color blue. Well, we have done a little work with, again, 2 species at a time. So this next image shows, again, that blue-green pigment, the Pseudomonas culture alone, and then the middle test tube just shows the uninoculated tube. But with Enterococcus faecalis, so E. faecalis, which is another bacterium that can be found it's a gut bacterium, but can be found in wounds or other infections. So this is a bacterium that can be found with Pseudomonas aeruginosa at a few different points on the human body. When we co-culture Pseudomonas aeruginosa with Enterococcus faecalis, we see that those test tubes are no longer blue-green. So this just like was absolutely fascinating. When the grad student came in and we'd started to chip away at this, and then she said, "Oh, by the way, they're not colored anymore." I said, "What? Well, you got to show me." So this led us down, a path that is one of our more recent papers. So that's also, a satisfying thing to have a result. But it seemed that what Enterococcus faecalis is doing is that Enterococcus faecalis is making ornithine. So if you've had to take biochemistry, or you've had other interest in how proteins are made, right? Perhaps you've memorized or even had to, draw out all the structures of the 20 required amino acids, right? But then you may or may not already know that there are additional amino acids besides those required to make a protein, and one of these is ornithine. And it seems that under most conditions, Enterococcus faecalis will make and leak some ornithine. And you could just add ornithine, which we did as kind of like the key to getting the paper published in our case. If you just add ornithine to Pseudomonas aeruginosa cultures, indeed, they turn less blue. But when you co-culture them, this is a condition where, unlike the Salmonella and Pseudomonas swarm plate assay that we were talking about a few minutes ago, Enterococcus faecalis and Pseudomonas aeruginosa will survive together in the same test tube or on the same plate. And it seems that at least one of the reasons that this happens is that Enterococcus faecalis will make some ornithine, and that ornithine, that blue pigment is one of the things that can kill off our cells or other bacterial cells. So it's one of the many ways, which add up to an arsenal of ways that Pseudomonas aeruginosa does, it just does mean things, right? So, yeah.
That's amazing about that communication. What is the I'm sorry, perhaps I misheard you. What is the ornithine doing?
Well, actually, we don't know exactly.
Okay.
We know that we know a bit that it's interfering in a pathway to make the alkyl quinolones. So Pseudomonas aeruginosa has more than one quorum sensing cascade. So it has a classic Lux system, acyl homoserine lactone, just like in those Vibrio's bioluminescent bacteria that you like to talk about, Mark. There are two cascades called the LAS and the REL system, but then there's a third cascade that is the Pseudomonas quinolone system. And what we do know, among the many things we don't know, is that adding an ornithine interferes with quinolone production. So that part of the cascade is somehow blocked. And again, there's a layer of complication there as well, along with some unknowns. But at the most basic level, we know that the ornithine is disrupting the cues that are needed to make piocyanin in the first place.
So and this is an important, because I bring this up, that this is not the same as quorum quenching.
This is not the same as quorum quenching. This is
Thank you.
This is stopping several points upstream of that before they ever made the quorum sensing molecules. So it's not making of those signals and then blocking them. It's like stopping it well upstream of that, somehow preventing a snowstorm, before you ever knew it was going to snow, right?
So what's interesting here for listeners and viewers, and this is how discovery works, and it's a matter of actually using probably the most effective machine in the laboratory, and it's not a multi-million-dollar Illumina sequencer. What it actually is your eyes attached to your brain.
Yeah.
And, and I several times in this podcast pointed out that the late science fiction writer Isaac Asimov used to say that discovery happens, and this is how Asimov was, is not some damp Greek emerging from the bathtub shouting Eureka. It's someone saying, that's weird. And that's exactly how this happened. Yeah. Because you knew what to expect from the organism. This is different, and you wanted to know what made it different.
Yeah. Yeah, that is exactly how that happened. Yeah, like that's different. It's a challenge and a joy and other things at different times, right? To get students at any level, undergrads, maybe even high school students, but graduate students and postdocs as well, to like, if you get something weird, it's not necessarily wrong.
Right.
It might be the interesting thing that's going to lead to a great paper for all of us, right? We need to follow up on things that seem odd. Yes, please be paying attention.
Yes, and the other thing is, this has happened to me over and over again, where I saw something that turned out to be really important, but I didn't think it was.
Yeah.
So pay attention to that,
right.
and it really pays off in a lot of ways. I often tell students, and they think I'm talking about tests, and sometimes I am. The term "I don't know" isn't a judgment; it's an opportunity.
Mm-hmm.
And the universe is full of so many unknowns, so many
Right.
things to study. This makes it so exciting. And I love that story, and I'm certainly going to link to that paper. That's wonderful. Thank you for sharing it.
Oh, you're welcome. Thank you for your interest.
Now, as I'm watching our clock a little bit, do you have a final experiment or observation that you wanted to share?
Sure. I think I would refer back to an older one. I was part of a group that discovered walking bacteria. So again, this is Pseudomonas aeruginosa, and it started as a collaboration with a good friend, Gerard Wong, who is in bioengineering at UCLA. And I was a new professor out of my postdoc, and I already knew Gerard. And he said, well, those time-lapse videos you showed of bacterial biofilm development, do you have more of those? And I said, well, I don't, but we could probably make some more. So I trained an undergrad in my lab, and he and I both set to work to record some more of these time-lapse images. And Gerard's thought was that he was advising a group of senior bioengineering undergrad students that needed a design project. He's like, well, we're going to modify tracking algorithms to track your bacteria. And realize this is now, over 15 years ago. So yeah. These days, that is very doable. But in those days, it was not trivial in any regard. So we sent off the data and thought nothing of it. And then he messaged me back, and he's like, well, there's some interesting things going on. Some of these bacteria seem to be cartwheeling. And then we can show the video that what we're seeing is that shortly after cell division, once we knew what we wanted to see, is that one bacterium will orient upright, and it'll use its type 4 pili, and it has to use multiple pili to do this. It appears to, walk, and in this case, it'll walk off the stage. It'll walk off the screen. But it all came as part of, some undergrads with a curious observation that a small percentage of them were actually short. So, you think of how microscopy works. It's basically all in 2D, right? Even things that we recreate to be 3D were a series of 2D. So the cells that were short, right? If it's a Pseudomonas cell, it's a rod-shaped thing, but the ones that were short were the ones that were sticking off the screen. So those were the ones that were oriented upright, and those were the ones that were going to be the walkers. So that's how we discovered all this. Through a series of iterations, and then, absolutely fascinating discovery, we were able to find that, Pseudomonas aeruginosa will use its type 4 pili to orient upright and then just walk around.
And what's interesting about all of this, and I'll put a link in the show notes to the original discovery about this, but the idea that and I use the term domesticate carefully, when you use laboratory strains that have been passaged in the lab, which most of us use, they absolutely have lost many things that they had out in their original environment. And it makes you wonder what things were missing when we look at typical model strains.
Yeah, that's a great point. We must be missing things, right? So that would be the argument for always going out every now and again to get some new out of a soil. Obviously, people do this from the clinical side all the time. They establish
Yes.
why the clinical strains might be different, right? They're not always different for any given behavior, but then sometimes they're radically different. So, and then, you could center on that too. We could go back to what we were talking about at the beginning of the episode. Like, you focus on that only and realize, well, all these clinical isolates are also not the same as each other. Each one is its own separate evolution experiment for some reason. And we're really left guessing, right? Let's be honest. We're guessing at what we think is why they evolved to be what they did. But it must be important if they've evolved to do it.
Oh, no, I agree. I mean, I don't, I hope geneticists won't be angry. I'm not sure we really understand selection. We can impose selection, but we don't understand all of its intricate effects.
Right.
And I really think that that's important to keep in mind. I mean, because we tend to impose our worldview on this, and they have their own agenda, and it's not ours.
Right. And we can do something like purposefully add an antibiotic, and then we know we added that. But did that change other things about the experiment that we're not paying attention to? And those probably influenced it also.
I mean, I cut my teeth on a predatory bacterium called Delovibrio, and my undergraduate professor, Sid Rittenberg, had been involved in the discovery of that, and he spent many years working on it, and he never made a frozen stock. Instead, he would passage it once a week for literally 20 years.
Right.
Until and that's why people who study Delovibrio don't use that original strain, 109J. And I'm curious how much its genome changed, but we don't have the original isolate, do we?
No.
So, but I believe that being optimized for life in the lab or life in glass or whatever it is you want to put for it. So that's why I think it's important to look at these unusual behaviors and see how they compare to, quote, wilder versions of the same organism.
Yeah. I think it's useful to consider all those things, right? It's useful to be practical and make a discovery. And most times any one discovery turns out to be relevant to one or more things. But then we could expand our view a little bit and say, oh, but it's not relevant to every strain we might see or every condition we might see. But if we don't start somewhere and ground ourselves, then we'll either go mad or we'll just not really ever figure anything out. So it's good to be a realist and realize the limitations as well as the advantages of any way we set up an experiment in the lab, right? That's just practical.
And science is iterative. You build on what's done before. So that's the power of it, in my opinion. I have to tell you, Joshua, this has been an absolutely fascinating podcast for me. And here's a secret. I do this podcast as much for me as for anyone who's listening and viewing, because I love hearing about this kind of thing. And I really appreciate your time in contributing to this podcast. Do you have anything that you would like people in particular to take away from our conversation?
Well, I think we can resort back to the it's complicated part. That certainly fits. And there's so many interesting things about what we do not understand yet about microbes and how they do things and how they interact. So I think we've touched on those points, but there's more left to discover than there is known. So
it's a wonderful time to be a microbial scientist. That's what it comes down to.
Yeah, for sure.
Well, thank you so much. I want to wish you and yours and your research team the very, very best.
Well, thank you very much, Mark. It's been a pleasure.
Thank you. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with, as always, epic links, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Joshua Shrout can be found in the Department of Civil and Environmental Engineering and Earth Sciences at the University of Notre Dame in Notre Dame, Indiana. Many thanks, as always, to David Renata for superb editing and Reaver Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.